So this week we are looking at experiment 16, which is the synthesis of triphenylmethanol. So this is a classic example of a Grignard reaction with a ketone. And the ketone's been carefully chosen so that the product is relatively simple to isolate. And this reaction sequence highlights this and develops the skills of preparing a Grignard reagent. We're preparing an excess solution of Grignard reagent to account for side reactions of the Grignard. And we are then going to react our generated Grignard reagent with benzophenone, shown here. So it's a highly reactive ketone. And after quenching our reaction with acid, we would be able to isolate triphenylmethanol. Because Grignards are oxygen and water sensitive, this reaction will be run under in oven-dried glassware, and the surrounding atmosphere above our reaction will be further dried using a calcium chloride drying tube. This can help minimize loss of our Grignard due to side reactions, but I would like you to note that the Grignard is used in excess in this reaction, and that's important for your limiting reactant calculations. Now, one unfortunate issue and one major sort of problem when we're looking at our product mixture from this experiment is that bromobenzene, when we generate phenyl magnesium bromide, can undergo a range of side reactions, either through protonation to generate benzene, or more importantly, through homocoupling reactions to generate biphenyl. This biphenyl is an appreciable byproduct that can be observed in our product mixture. And our goal in this lab is to synthesize triphenylmethanol and then separate triphenylmethanol from biphenyl via trituration. So that's our first overall goal for this lab. So initially, we'd have, when we run our reaction to completion, we'd have a mixture of our triphenylmethanol and our biphenyl. From there, our reaction mixture is initially in diethyl ether, and the first So the mechanism for this homocoupling reaction, so there are a few ways to think of this. So during generation of your Grignard, so the main issue for this homocoupling side product is mainly that once you generate Grignard, if you have any of the bromobenzene floating around, the bromobenzene in our Grignard can react in order to generate this biphenyl. So let me just make sure that I'm drawing the mechanism appropriate for this given course context. So I'm trying to think of a reasonable way to draw this in a way that we're not going through excessive organometallic mechanisms. The best one way that you can think of this process is your equivalent of Grignard can self-react with your equivalent of bromobenzene. So you can have, give me one moment to find a suitable mechanism for this case. Because it's often tricky to determine whether we're dealing with a radical mechanism or whether it's a formal cross coupling. Give me 1 second. So give me one moment. The mechanism is often not particularly well defined for this homocoupling step. I'm just trying to think and trying to see in the literature whether it's drawn as a radical or a formal like insertion, oxidative addition, reductive elimination mechanism. So these are some possibilities. So when you have your phenyl magnesium bromide generated, and you have your alkyl bromide, so one potential option that would lead to homocoupling is where you would end up with sort of fishhook arrow dissociation, make a carbon-centered radical. From here, your alkyl halide can react with the magnesium radical, and then your two alkyl radicals can couple. That's one plausible method to generate biphenyl. From what I've seen, oftentimes when you see these oxidative couplings involving Grignard reagents, there's often an external reagent, such as either trace iodine found in your reaction mixture or trace amounts of another metal that's helping to facilitate this oxidative coupling process. I can look into this in a little more detail and I can come back with some more definitive mechanisms for this specific substrate under these conditions. Generally, this kind of homocoupling occurs when you have an excess of Grignard and you still have unreacted alkyl halide. Yeah, this would be fine for the mechanism section. I'll look into the literature to see if this has been fully defined and if we have a full mechanism for this section. It's often tricky since a lot of the literature on this is based on sort of taking a Grignard and then using further additives in order to facilitate this coupling process. I think it's most likely going through a radical mechanism. Okay, so continuing on. So upon reaction of our benzophenone with phenylmagnesium bromide, we isolate our mixture containing triphenylmethanol and biphenyl in diethyl ether. And our first step, considering that our solution is currently basic, we need to acidify our reaction mixture with hydrochloric acid. And our goal in this case is to break up our insoluble inorganic salts, and in order to fully quench all of our excess Grignard and excess equivalents of base present in our reaction mixture. So we're going to add hydrochloric acid until we see a litmus red solution. Now, hydrochloric acid is an aqueous solution. We initially have diethyl ether as our reaction solvent. So when we look at the layers for this separation and extraction, the ether layer will be on top. So we're going to collect the diethyl ether layer, and then what we're going to do is we're going to extract the aqueous layer with diethyl ether in order to remove any trace amounts of organic compound still remaining in the aqueous layer. This is a textbook method of extraction. So in this case, we're extracting out any residual triphenylmethanol that is found in our aqueous layer. Does this idea of separating our organic and aqueous layer and then further extracting the aqueous layer to get all of our organic compound out of the aqueous layer, does this idea make sense to everyone so far? Okay, so once we've extracted the aqueous layer, we're then going to collect all of our organic layers in one single vial, and we're going to rinse with cold water. The point of this step is to remove the residual acid before concentration. It's very problematic to concentrate reaction mixtures with any trace acid or base, as you can often see decomposition side reactions. So we're rinsing with cold water to remove the trace hydrochloric acid residue. And from there, we're going to have our combined ether layers. With our ether layers, which contains our reaction product mixture, we're then going to load our sample. So we're then going to load our sample into a Craig tube. This is used for recrystallization and trituration, and we're going to record the mass of the empty Craig tube before we've added our reaction mixture in diethyl ether. We're then going to evaporate the reaction mixture and we're going to isolate our crude solid. This mass of our crude solid is really important to consider, and it's really going to help us begin to understand, once we separate out the byproduct, how much of this product mixture is our desired product and how much of our product mixture is the undesired byproduct. So this product mixture is not pure. It contains biphenyl. So what we're going to do is we're going to purify with ligroin, also called painter's naphtha. It's a non-polar, it can be aromatic as well, solvent. Painter's naphtha primarily contains hexanes and other long chain hydrocarbons. I'd like you to think of ligroin or painter's naphtha as a very, very non-polar solvent. To answer the question in the chat, yes, you weigh the crude product mixture first. That's an important mass to note. It's always a convention before purification for you to weigh the crude product. Okay, so when we're purifying, yes. So when we're purifying our product mixture via trituration with ligroin or painter's naphtha, let's look a little bit as to how trituration works. So fundamentally, trituration is selectively dissolving impurities that are absorbed to our solid or trapped within our closely packed solid. So we're going to be agitating our reaction mixture and thoroughly mixing our insoluble solid product with this immiscible solvent, in this case, painter's naphtha. The goal of this process is to selectively dissolve our impurities and to break up our solid so the solvent can efficiently dissolve and separate out our non-polar impurity, which in this case is biphenyl. So the reason why this trituration works is if we compare triphenylmethanol and biphenyl, which of these two compounds is the least polar? Which of these two compounds is most similar to painter's naphtha? Which of these two compounds would be more soluble in a very non-polar solvent? And I see in the chat a lot of students are mentioning biphenyl. That's exactly correct. So this nonpolar solvent will selectively dissolve your biphenyl during trituration, and that in turn would allow you to separate out this biphenyl through repeated trituration of our solid. So rinsing the solid with a nonpolar solvent removes the biphenyl and other nonpolar impurities. So we're going to be taking our solid, adding our solvent, rapidly agitating our solid to generate a very fine suspension. And then in order to separate out our solid and liquid during trituration, we're going to utilize the Craig tube fully. So the Craig tube has a cap that goes on top of the tube. And essentially what we do is we load the Craig tube with the cap inverted in a larger conical tube. And when we centrifuge our tube, the liquid will be forced through this gap while our solid remains trapped in the Craig tube. This method of trituration followed by separation using the Craig tube allows us to rinse our solid with a immiscible solvent and allows us to separate out the liquid mixture from our solid. So we're going to repeat this process for a total of 3 times. And at the end of this process, we're going to be left with our cap with a large amount of solid deposited and our corresponding liquid layer. The liquid layer will be concentrated and the mass will be used to determine the mass of the byproduct, or byproducts to be exact. The solid is purified triphenylmethanol. And so once we have our collected solid after trituration, we're going to be able to record the mass of isolated purified triphenyl methanol, and we'll be able to collect an IR, NMR, collect a melting point, and report the yield. So this is relatively boilerplate analysis. Triphenyl methanol is not appreciably soluble in painter's naphtha because the hydroxyl group makes it slightly more polar. The difference in retention factors as well points to that fact. There's a substantial difference in polarity and solubility due to the hydroxyl group. As we discussed in lecture, the hydroxyl group is a relatively polar functional group. Okay, so let's talk about what's new in this lab in terms of what new techniques that we're using. So one technique that we're going to use to get a sense of how many different components are in our product mixture and how effective was our separation at separating out this biphenyl from our desired triphenyl methanol. And the method that we're going to use is thin layer chromatography. So thin layer chromatography, in essence, relies on spotting your sample, and you can make as many lanes as you want containing your reactants and products or just different mixture components. And the main thing to note is on this plate, if we zoom into this plate, each of these TLC or thin layer chromatography plates are coated with silica gel, which you can think of as a surface containing multiple hydroxyl groups. So your compound can interact with your stationary phase, and it will interact with the mobile phase, which is the solvent that is slowly moving up your TLC plate via capillary action. Now, just like when we discussed column chromatography in the context of gas chromatography, based on your compound polarity and structure, it will interact more or less strongly with the stationary phase. Further, more polar compounds generally react stronger with the stationary phase and move less readily up the plate. So if we took like snapshots before, during, and after, so if we separated a mixture of two components, A and B, and we ran A and B up the plate. If A is more polar, it will move less readily up the plate compared to non-polar compounds such as B. At the end of our separation, we'll have a series of spots. Each unique spot indicates a unique compound, and compounds with the same location up the plate have identical polarity, and in many cases, if the two spots have the exact same retention factor, then that tells us that the two spots correspond potentially to the same compound. Now, one way of categorically logging the distance a spot moves of the plate, and one way of logging the relative polarity and how readily compounds progress up the plate in different solvent systems is using the RF value known as the retention factor. The retention factor is calculated first by measuring the baseline and the solvent front and marking those. And the first thing that we're going to measure is the distance from the baseline to the solvent front. How far does the solvent move? And we're going to write that number down. Next, we're going to measure the distance from our baseline to our spot. So the RF is the distance of our spot over the distance of our solvent front. So suppose distance A was 5 centimeters and distance B was 18.5. You're going to need a ruler for this lab, and you'll see some screenshots posted from this lab on Canvas containing the TLC plates for you to analyze. Now, from each of these distances, we can calculate the RF for each of our two reaction mixture components, or each of the two spots that we see. And then from there, we can compare to the literature values found in your lab manual for this solvent system. So if you reference values in the lab manual, you're going to need to cite the lab manual fully. Does this make sense to everyone so far? So thin layer chromatography is a method to separate reaction mixtures on a very small scale. It's not used preparatively in this case. It's just used to get a sense of how many things do we have in our product mixture, and before and after purification, were we successful in our purification? So, for example, if we have our mixture of triphenylmethanol and biphenyl, initially we would expect to see two spots, with the lower spot, or the more polar compound, corresponding to triphenylmethanol. Now, at the end of our purification, we are going to take another TLC of our product mixture, and we're going to look and see how many spots, how many compounds do we have in our product mixture after purification, and did we successfully isolate the triphenylmethanol? So by looking at the TLC plate before and after purification, it allows us to get a sense of whether or not our purification successfully separated our compounds. Does that idea make sense? So typically RFs have a small amount of variance and they depend on the solvent system. So generally RFs are measured for a specific solvent system, and identical compounds can have the same RF. It's not a perfect assignment. That's why we collect the other data to further support our argument that the sample is pure and that the sample is our desired triphenylmethanol. RF is just the RF value, and taking a TLC is just a very quick method to immediately see how many things are in my mixture, how different are they in terms of polarity, and what kind of column separation can I use. Does that answer your question? Well, you can calculate a percent difference compared to the literature value for your RF. You generally see RF values that are relatively close, but there are always going to be some small differences, depending on the TLC plates that you use, depending on the solvent system that you use, and depending on how large your spots are. There are always some variance. Does that answer your question? Perfect. So that is a full overview of the laboratory and the different purification and separation methods we're going to be utilizing as part of this laboratory. There's a lot of data for you to analyze, so you want to make sure you have all of the literature, spectra, and physical property data required in your pre-lab. So with that said, for the remainder of this lab session, we'll be working on our pre-labs and lab reports, and I'll be happy to look over any report drafts and provide feedback on your calculations and reports.